Hello? Yeah. Hello?
சார் என்ன ப்ரெசன்டேஷன் போட்டு பார்க்குறீங்களா உங்களது பிவிஎஸ்?
ஓகே, நான் ஸ்டாப் பண்ணிடுறேன் சார்.
நானே I will start the presentation.
ஓகே,
நானே பண்றேன் பெட்டர்.
ஓகே, ஆமா.
ஓகே, நான் இது ஸ்டாப் பண்ணிட்டுமா சார்?
ஓகே, ஓகே. அவரது ட்ரை பண்ணிட்டேன்.
ஓகே,
ஓகே, நான் இப்போ இது வேணா ஸ்டாப் பண்ணிடுறேன் சார்.
ஓகே.
Okay, so I will start my presentation now. Entire screen. Is it visible, sir? Hello?
இது ஸ்லைட் ஷோ மோட்ல போடுங்களா?
போடுறேன். Is it visible?
விசிபிள்.
Okay. Right. Okay, sir.
அந்த கீழே வந்து அந்த இது இருக்கு, ஹைட் பண்ணு. ஓகே, வெரி குட் சார். ஓகே, let it be there and another few minutes formal introduction will be taking place, then you can start.
Right, sir.
No, just 2 minutes இருக்குதானே?
So I'm going to start now or in another 2-3 minutes? Hello? Hello? Hi, sir.
Now we'll start in 2 minutes.
2 minutes. No, that's why. Just wait
Yeah.
for me for a minute. Huh? I will be back.
Yeah.
Yeah.
Okay. Okay.
Yes, sir. I'm back. Huh? Yeah. Is my
Yeah.
audio volume is okay? Yeah.
Fine, sir. Fine.
Right.
Fine.
Yes,
Thank
sir.
you.
Yes, sir.
Actually, recording start.
Shall we start?
Yeah, recording. Let the recording start. Okay. Okay, we can start now.
Yes, sir. Good evening, everyone. On behalf of Good evening, everyone. On behalf of IEEE CS Madras, ACM Chennai, and CSI Chennai chapter, I welcome you all for the webinar on applications of machine learning in life sciences. As we all know, today's world has huge, large amount of data in the field of life sciences related to drug discovery, microbial phenotypes, generation of diseases, specific medical images, etc. On all these data, a powerful approach which can decipher complex hidden pattern need to be identified, where the ML supports to provide all these pattern recognition. So we decided to have this presentation on applications of machine learning in life sciences. Machine learning and deep learning methodologies have augmented and relational process of drug discovery. High performance ML algorithm have it make it possible to rapidly adverse the data and to analyze the multimodal and multidimensional data sets dealing with precision medicine. Additionally, ML-based workflows type found immense applications in life sciences. So, to put forth this beneficial webinar, let us identify the eminent speaker, and we let it use the speaker now. Dr. Jayaraman had done in the bachelor's and master's degree in Chennai Chemical Engineering from Madras University on doctorate in chemistry from Pune University. He had been associated with National Chemical Laboratory Pune, a consistent laboratory of the Council of Industrial and Scientific Research CSIR, India since 1976. Starting as a junior scientist in the Chemical Engineering Division of the National Chemical Laboratory Pune, he worked with currently Dr. Jayaraman is visiting professor at Nadar University and a distinguished professor at Flames University. The research area of the interest include modeling and simulation, optimization and application of machine learning and artifacts intelligence to different disciplines, including core computer science, process fault detection and monitoring, information security, Chemo and Bioinformatics. Dr. Jayaraman has taught several courses, including computational biology, 100 papers in the reputed international journals to his credit. Welcome you, sir. I also welcome our associate partner, IEEE CS Chair, Dr. P. Sakthivel, and ACM Chennai Chair, Mr. Hedar Mohan, to the webinar. I also welcome all our past members, present members, past OB members, present OB members of all our professional societies. I also welcome all the enthusiastic participants, and I appreciate the participant to join this wonderful webinar. Thank you all, and let me leave the podium to our speaker. Welcome, sir.
Good evening. Thank you very much. I first sincerely thank IEEE, Chennai, and all the associated associations for giving me an opportunity to talk before you. I'm going to talk on the applications of machine learning in life sciences. Yeah. The recently implemented next generation sequencing methodologies has provided sequencing of genomes very fast, and improved algorithms in conventional machine learning, coupled with advent of deep learning methodologies and high precision instrumentation in biology, has made a very great impact on the advancement of life sciences. In this webinar, I'm going to provide initially a small introduction to machine learning and bioinformatics, so that this particular lecture or webinar is going to address all sorts of people with very little knowledge of both ML and bioinformatics. After the introduction, I will briefly discuss some of the applications. Yeah. As we know, DNA is the genetic material within the nucleus. Replication of DNA creates new copies of DNA. Transcription creates an RNA using the DNA information. Further, the translation creates a protein using the RNA information. So the DNA consists of 4 nucleotides: adenine, thymine, guanine, and cytosine. The nucleotides are arranged in a double helix structure, and a DNA sequence looks something like that. A given DNA sequence consists of a specific arrangement of the 4-letter words A, T, G, C. Different DNA sequences will have different arrangements, different of this sequential pattern, and different lengths also. Now, a protein sequence or a peptide consists of a linear sequence of 20 different amino acids. They can be represented by a single letter or 3-letter code. A protein sequence, as I said, again, is a distinct combination of these 20 different amino acids, and then different protein sequences can have different lengths. So the protein structure can have specifically helix, coil, and sheet-like structures, and different protein sequences have different structures. Yeah. So this is 20 different amino acids. Now, genes and genomics. Genes are part of the DNA sequence which carry the information that determines our traits. Each cell in the human body contains around 25 to 35,000 genes, and genomics is a study of all the person's genes, including the interactions of the genes with each other, with proteins, and with the person's environment. Machine learning methodologies can identify genes which code for proteins, the functions of genes, and protein functions. NGS methodology can sequence the entire human genome in an hour. So this has generated a lot of new genomic information, which has produced large amount of data. So ML has facilitated processing of this data into useful algorithms for the benefit of the mankind. So a protein sequence can be both soluble and insoluble. If a protein becomes insoluble, it has implications in cancer. So given a set of sequences, we can identify whether a sequence is going to be soluble or insoluble. So so machine learning, different kinds of classifiers can classify a given set of sequences into soluble sequences and insoluble sequences. Now, let us give a basic definition of machine learning. Machine learning is the ability of computers to learn from data, which identifies the patterns which are hidden by complexity. And a computer program is set to learn from an experience with respect to some task and with a defined performance measure like accuracy, say, for example. Machine learning derives its strengths from different fields of science and engineering. It is definitely a subset of artificial intelligence, where artificial intelligence consists of both logic, rule-based methodology, as well as pattern-based methodologies. Machine learning majorly depends on pattern-based methodology. It also derives its strengths from information theory, statistics, neurosciences, and computer science, and also operations and optimization and control. Yeah. So let us give a very brief methodology of machine learning with a small example. We can identify people who are fit for army. Army has got a historical data of people who are fit for army and who are not fit for army. So there are many different domain attributes or features which they use to identify people who are fit for army. So let us take simply two different attributes so that we can understand what is machine learning. They have the data of height and weight of people who are eligible for army or not. If we plot this data, see, we have color coded it as blue and red because we already know from the historical information that the blue color people are fit for army and red coded people are not fit for army. So for having put this into this graph, you can see it clearly separates into two different groups. The algorithm here is the line which separates the data into two different groups. So this is a simple linear classifier we have explained. Now, when we have done this with the training set of data with historical data, when a new person comes, if you just put it in the coordinates into this graph, if it comes here and settles above the line, we say he is selected for army. And if he has got the weight and height, and if it comes to the coordinates comes and settles below the line, is not fit for army. The learning here is to identify the right line. Different lines can separate the data into different groups, but only the best possible line with best values of slope and intercept separates the data perfectly into two different groups. This is, yeah. In biology also, we have so here we have output. This output is nothing but selected for army or not. That has a label 1 and 0. 0 is not selected, 1 is selected. It's a function of the inputs. The inputs are weight and height. So this algorithm, now we can maps the inputs into output. So you get a functional relationship between the weights and heights and the output, whether it is selected or not. Yeah. In biology also, we have got functional relationship between inputs and outputs. The inputs are the sequence properties. It can be protein sequences, DNA sequences, gene expression profiles, and multiple omics properties, structural properties, etc. The input can be drug properties. The output can be whether it's going to be active drug or inactive drug. So given a set of input properties, we can identify whether a person is going to be a normal person or a disease patient. So percentage of genes in a human genome is only 1.5%. So identification of genes which code for proteins is a very important task. It's a gene annotation task. Given a set of DNA sequences, we can identify which of them are genes, which code for proteins. Different machine learning methodologies facilitate this identification and classification job. So here, given a set of input sequences, DNA sequences or structure, a machine learning algorithm develops a model, it learns, and then it can be used to predict. Once it predicts, the prediction can be used to experimentally annotate new genes and then identify and then use this predicted gene sequences, the input data, and then you can improve your algorithm for better prediction. Yeah, more the data, the merrier. Yeah. There are essentially 3 different types of learning methodologies. One is the supervised learning methodology. It learns the functionality from both input and output data. Unsupervised learning data, we do not have any output data. Derive the structure from input data alone. Whereas reinforcement learning methodology learns from experience, whereas supervised methodology learns from data. So there are different algorithms which have been developed in machine learning for different kinds of methodologies, supervised, unsupervised, and reinforcement, and they have in the supervised learning methodology, support vector machines, logistic regression, decision tree, random forest, and k-nearest neighbors are some of the algorithms which can classify data into different groups, which can identify different functionalities of genes and proteins. And linear regression, kernel regression, again, is a supervised learning methodology where we have data. The output is a continuous output, like activity, prediction of drug activity. And multiple functions can be identified. Unsupervised learning methodology, again, derives structure from the data without any labels. It can be used for dimensionality reduction as well as data visualization and clustering. Multiple algorithms are there for clustering and data visualization. The usefulness is to group the genes expressions to different groups, and then phylogenetic analysis of multiple sequences and visualize data. Reinforcement learning also can be used for multiple purposes. Yeah, come to the pipeline later. We will normally use machine learning when we have patterns in the data, even if we don't know what they are. And if we don't have a functional relationship mathematically, then we use machine learning as a data-driven modeling approach. So in genomics, the use is identification of gene functions, protein functions, and disease prediction. This is just a sample. Yeah, we'll just again give so given a set of sequences, we can identify gene sequences. We can identify sequences into essential and non-essential genes. Essential genes carry out different essential jobs, and it's very important gene identification problem. A set of sequences, we get the attributes here, the input domain information we have extracted is the frequency of different letters, nucleotides, A, C, A, T, G, and C, and different patterns of frequencies. The blue color sequences with certain frequencies of all these 4 nucleotides are essential genes with target 1, and the maroon colored sequences with the different patterns of combinations of the nucleotides are non-essential. So we know this a priori in supervised learning algorithm from experimental annotation, both the inputs and the outputs. So with this, again, let us take the example of the frequency of G and frequency of adenine. Now that learning is, so if you put all these examples here, you again see you have to identify the best possible line which separates the data into 2 different groups. Here again, the learning is the equation of the line here. So once we learn this again, new genes extract the information of the frequencies of nucleotides, put it in the graph, and then if it falls above the line, then they are essential genes. Fall below the line, it's non-essential genes. Yeah. In some unsupervised learning algorithm, on the other hand, we only have the input data. We don't have output class labels. So as an example, given a set of sequences, we can extract the domain information in terms of these two physicochemical properties. As I said, multiple attributes can be derived from DNA and gene and protein sequences. Let us assume that we have extracted two attributes, hydropathy and net charge, from this set of sequences. Once we have done this, we only have for these sequences this information. Now, with this information, we can use an unsupervised learning algorithm to cluster the data into two different groups. Once you have clustered the data into two different groups, examples belonging to having certain functionality will definitely cluster into a same group. So let us go to each cluster, and these 5 examples clustered into them, we can experimentally annotate and see what sort of similarity they have. As an example, these sequences have to possess allergen properties, whereas the sequences which have grouped into another cluster have been experimentally found to be having no allergen properties. So here, unsupervised learning methodology, we derive structure and information after clustering. Okay, this is the main difference between supervised and unsupervised classification methodology. Principal component analysis is again an unsupervised learning methodology which makes coordinate transformation to capture the most informative attributes, small number of attributes from a large dimensional example. So it is used to reduce dimensionality. Again, the TSNE is a T-distributed stochastic neighborhood embedding methodology. Again, reduce dimensions. It does by a different methodology by minimizing the difference between the distributions of the original and the reduced dimensions. They can be used for visualization. Data visualization in computation biology and life sciences is a very important methodology. Different softwares are available for visualization. Here, from that cell types, different from hundreds of features, PCA reduces the dimensions to 2 dimensions with PC1 and PC2. And these 2 dimensions have captured the essence of information. And with these 2 dimensions itself, we are able to differentiate between the different cell types. Similarly, the stochastic neighborhood embedding visualization is able to capture information about multiple disease types of persons. Yeah, with only 2 dimensions. And hierarchical clustering, again, is an example of dimensionality reduction to capture phylogeny information of sequences. Yeah. So in the example where I've used for unsupervised learning, we have extracted hydropathy and net charge as a domain information. And domain information can be provided in multiple ways. For example, in protein sequences, you can pass the numerical representation of the sequence itself. In deep learning, we can provide the sequence information as it is. And we can also extract the 20 different frequencies of the amino acids, and then 400 different 2-letter combinations, and 8,000 different 8,000 different 3-letter combinations, and higher order frequencies also. We can get homology information in terms of blast profiles, remote homology information, we can get motif information. Now, as I said, proteins also have secondary and tertiary and quaternary structures. You can get structural information. You can extract multiple physicochemical properties and structural features like surface accessibility, the coordinates of the atom, etc. So all these now represent large amounts of domain descriptors, and we can use machine learning methodologies itself to identify the most informative descriptors, which is known as a pre-processing method, which is feature selection. Yeah. So this is one cartoon where a secondary structure of a protein has 3 different types: alpha helix, beta sheet, and random coil. Yeah. So I will now go to the applications. Yeah. Is this? Are you able to see this? The second set of slides, life science applications.
Yes, sir.
Okay,
Yes, sir.
thank you. Yeah, okay. So we'll go to the life sciences applications. There are several applications in different fields. Applications are mostly first in the development of the bioinformatics itself, in the genomics, proteomics, and multiple omics, and then drug discovery and other applications. Let us start with the genomic applications. Genomic, as I said, is a field of study of the genes and interactions. ML application can be employed to annotate genes, to find genes from set of DNA sequences, because genes only code for proteins, and then expressions of proteins is a very important factor in identification of diseases. Then phenotype predictions, like given a set of input sequence attributes and structural attributes of genes, proteins, and other properties, we can identify whether cancer, different protein functions, properties like druggability of non-druggability of drug targets, and we can use multiple classifiers in machine learning with different attributes for genomics applications. Now, GWAS is a genome-wide association studies. It's an approach which involves rapidly scanning the markers and getting the information about genetic variations of different kinds of people, and getting the single nucleotide form polymorphism, wherein we can identify mutations of the gene sequences at different locations of a given gene or multiple genes across the genomes of different people. And this information can be employed as input attributes to a machine learning algorithm, which can identify whether these combinations of mutations can be associated to a particular type of disease or not. Yeah. We have a protein function annotation problem, which is also comes into the genomics. We have in around 2006 employed a support vector machines based methodology for predicting the propensity of the protein to be soluble or to form inclusion body, which is going to be insoluble protein. As I said, identification of protein which is going to be soluble or insoluble has implications on predictions of cancer. So let us explain what is support vector machines. So before that, we have employed different sets of attributes, which includes concatenating the 20 different amino acid alphabet into a reduced number of alphabets, depending upon certain physicochemical properties. And then we also used the typical conventional compositional properties. We also use hybrid modules coupled with certain physicochemical properties. In total, we have about total of 150 attributes, out of which we selected about 135 attributes. Yeah. So the problem here is given a set of protein sequences, and from experimental annotation, we know certain set of sequences which are green color coded, we know they are soluble. So other set of sequences which are maroon coded are insoluble. So with these two classes of sequences, we have extracted from the sequences multiple properties, which multiple attributes amounting to 135 attributes. And then we have tried to build a classification model to separate the data into two different groups. Once we use this classification model with existing set of known proteins with known functions, when a new protein sequence, we can use this model to identify whether a given sequence is going to be soluble or insoluble, which provides very valuable information for disease prediction. So support vector machines is introduced by Vapnik and co-workers. It's a very high performance classification methodology used in different fields of science and engineering. SVM, what it does is it uses a maximum margin hyperplane. Now, if you have only two attributes, it's a line. If you got three attributes, it becomes a plane. If you got multiple attributes, like we have 135 attributes, we have to use a hyperplane. So it uses a hyperplane, which is maximum margin, means it tries to produce a hyperplane which maximally separates the closest example belonging to two different classes. This margin it tries to maximize. Once this is done, New examples can be predicted by using, just 1 minute. Sorry. Okay. New examples can be predicted with a certain amount of confidence because it gives some allowance for mistakes. Yeah. Now, not all examples can be classified by using a linear classifier. As far as example, for this particular set of examples, now we have to identify the quadratic surface. Chessboard is an example where you cannot use any line to separate the black and white squares. Likewise, this set of examples, you cannot use any line to separate the data into two different groups. Our solubility problem also falls into a non-linearly separable example. Multiple methodologies can be used. Different classifiers use different methodologies. Neural network uses a set of non-linearly activated transformed neurons to handle this nonlinear separation, whereas SVM uses a very elegant methodology by taking the data which is not linearly separable in the original set of attributes. In the 2-dimension, it's not linearly separable. It takes the data into a higher dimensional attribute space and uses a linear classifier to separate the data. This can be identified as, if people are sleeping, you can't identify by using any line people who are having a height more than a particular height, say for example 5 feet 7 inches. Now, if they stand up, now we've got an additional dimension is introduced in this 3-dimensional, this thing we can now see. With these 3-dimensional coordinates, we can identify people who have a higher height. Yeah. Now, some examples of gene and protein functions. These are just a sample of examples. Tissue-specific gene expression is a very important methodology so that you can identify genes which are wrongly expressed, and then you can use them as targets for developing drugs. And prediction of antimicrobial peptides, which we have done some work, we will explain it later. Identification of protein binding sites, again, implications in drug discovery. Localization of proteins in different nuclear compartments and cellular compartments. Identification of proteins which are toxic, like that. Yeah. We also have another SVM methodology to identify binding pockets of glucose by using SVM and molecular dynamic simulations. Yeah. So next we'll go to the applications of machine learning in immunoinformatics. Yeah. Antigens are recognized as foreign substance by human body, and they stimulate an immune response. Our immune system recognizes the specific 3-dimensional shapes of antigenic regions of foreign bodies, which are known as epitopes, and these are the antigenic determinants. An epitope is able to bind to or interact with a specific antibody of our host cells. Now, these antigenic determinants of the virus or other foreign cells have different shapes, and this can be identified if you can identify these different shapes, and these are the epitopes. And if you can predict this epitope, it can be used for reverse vaccinology. Yeah. Yeah. So machine learning algorithms can use methodologies to identify epitopes which has potential to reveal newer binding sites in a variety of antigens for the development of vaccines. This is reverse vaccinology. There are also tools available for predicting the antibody class to which the epitope can potentially bind. ML has been applied to so many other areas of immunoinformatics also. These includes tumor antigens and cancer biomarkers, toxin prediction of antimicrobial peptides, and ML-based methodologies have also contributed significantly in tackling the recent COVID pandemic. Yeah. Recently, in this month, we have employed subautomations-based methodologies to predict the epitopes which can bind to different antibody classes. So again, it's a multi-label classification algorithm. I will not explain more than this because we have got more methodologies to explain. Yeah. The next one we are going to explain is the conventional neural networks and the deep neural networks. because neural networks and deep neural networks have again been used in life sciences in different applications profusely with high performance algorithm. The neural networks, it mimics the working of the neurons in the brain. The inputs are inputs like we have the inputs like the protein sequence features like hydrophobicity and charge and different properties as input features. The input features are weighted and nonlinearly transformed by using some activation function by different neurons in the hidden layer, and then it goes to the output and compares the values of the actual output, which we know belong to two different classes, and any given sequence can predict to whether it can be a soluble protein or insoluble protein. So the input features which have been weighted and nonlinearly activated by different layers, whether if it predicts correctly the output, then it's fine. Otherwise, we backpropagate them and then reduce the errors and then readjust the weight. And this is iteratively done to finally get the best possible performance with least error. Now, if hidden layers, if we got... Hello? Yes? Hello? Is there anything? Hello?
No, sir. You can proceed. No problem.
No problem. Okay, right. Yeah. So, if neural network has got a small number of layers, it can have one layer, two layers. Normally, normal jobs, a two-layer neural network can handle. And if there are images and large amount of data, you have to use multiple layers, multiple hidden layers, which is now known as deep neural networks. Yeah. Deep neural networks and deep neural networks use different activation functions like sigmoid, hyperbolic, rectilinear, ReLU, etc. The ReLU is normally used in deep neural networks, which has some specific properties. And we also use sigmoid as well as hyperbolic tangent units in deep neural networks, depending upon the particular problem at hand and the situation and the position at the networks. Yeah. As an example, this epitope data set, IEDB has got a set of epitopes which exceeds around 20,000. So this particular paper has used 20,000 positive examples from this epitope database, which is a very powerful database, and also used negative examples which are not experimentally annotated to be epitopes. With these positive and negative examples, it uses a deep neural network with about 4 layers. And the input is taken as the 400 different dipeptide frequencies of the epitopes, and then uses the deep neural network successfully to classify the data into epitopes and non-epitopes. So the next field is proteomics. Another important level of organization of biological systems is the proteome. In proteomics, we can, using experimental methodologies like Lstms methodology, liquid chromatography, mass spec method, we can quantify different proteins and peptides and profiles. This profiling of the protein quantification can be used as input in machine learning methodologies to identify functionalities. For example, the protein quantification profiling has been used to identify and distinguish between different causes of chronic kidney diseases. It has also enabled identification of prognostic markers and to distinguish severity and non-severity of COVID-19 patients. And we know these prognostic markers are different expressions of proteins like D-dimer and CRP levels, and multiple markers have been used apart from this to identify severity on non-severity of COVID-19 patients. Yeah. This is one cartoon of how the protein profiling has been put into the deep neural network to identify particular disease condition, severity of the disease conditions. Yeah. The next application is in terms of metabolomics. The experimental techniques for metabolomic studies can accurately quantify the different metabolic products like amino acids, fatty acids, carbohydrates, and other products of cellular metabolic functions. If the metabolic functioning is not proper, this pattern of the amino acid quantities can differ from the normal metabolic patterns. So this metabolic quantification of the products can be used as input and domain information into many different machine learning algorithms and can be used to predict the phenotypes of the diseases. Yeah. Now, we have individually explained the genomics, proteomics, and multiomics methodology. Now, all this omics data can be gathered together. And when we gather this multiple omics data, it really becomes a very big data problem with multiples of descriptors. So all this omics data can be combined together and then can be used as input to the, essentially because we are having deep multi-omics data, we cannot use the normal categories of classifiers. We would use deep learning methodologies and input this multi-omic data to identify markers associated with the different diseases, including prostate cancer. And we can use this different combinations we can use of the different omics data. So you can quantify transcripts, the protein profiling, and metabolites and lipids to predict the clinical severity. We can also use DNA methylation data, which is epigenetics data. The epigenetics is a very important genomics information. Epigenetics determines the epigenetics can modifications can have a great impact on the disease conditions. Now, the sequences, DNA sequences can be methylated, and this methylation is an epigenetic modification. This epigenetic modification doesn't change the DNA sequences, but it has great implications. This methylation has got implications in cancer studies. So the epigenetic modifications along with gene expressions and mRNA data can be combined with deep learning approaches to identify different markers associated with cancers. Yeah. Next, we go to the applications in drug discovery. In drug discovery, there are 4 different stages: identification of target and validation. Compound screening and lead discovery is essentially a chemoinformatics methodology. Preclinical and clinical developments. Yeah, the drug target has to be relevant to the disease phenotype, must have a good therapeutic window, and should not have any side effects and should not disturb the normal functioning of the target. Yeah, so in multiple stages, machine learning methodologies have been used in target identification and prioritization of ranking based methodologies for using gene disease associations, whether a target is going to be druggable or not. So given a set of targets, we can use machine learning methodologies to identify targets which are druggable or not targets, use this supervised learning methodology and extract the attributes of the new targets and see whether it is going to be druggable or not druggable. Yeah, as I said, screening and lead discovery is a chemoinformatics methodology. They are also used in preclinical development, clinical development, in essentially identification of tissue-specific biomarkers, drug response signatures, prediction of biomarkers of clinical endpoints, and finally, in the clinical development, the response of drug by different cellular phenotyping in oncology, precise measurements of the tumor microenvironment immuno-oncology. Also, we can, in the clinical development stage, it is used in multiple different applications apart from what is given here. So, yeah. The other applications of drug discovery is SVM. One particular application where SVM has been used to classify proteins into drug targets and non-drug targets for breast, pancreatic, and ovarian cancers. Machine learning can also predict cancer-specific drug effects, can identify antimicrobial peptides. We have employed support vector machines methodology to identify peptides which have antimicrobial action, which includes multiple activities like antifungal activity, antibacterial activity, and antiviral activity. So this resource is available now, even now. So in this resource, it has a database. Also, it has got a prediction server. You can give any given peptide sequence, and then it will predict its antimicrobial activities. Yeah. Okay. So we'll not talk about biomarkers. Yes. The next application now will go into medical imaging and signal processing applications. Medical imaging like X-ray images and MRI and then CT images. You can extract information directly from the images, or you can use images themselves from the deep neural networks and utilize it for classification of different diseases. And here, for example, deep learning approaches based on CNN have been utilized for classification of lung diseases, employing CT images, and tuberculosis manifestation has been analyzed by using X-ray images. So the convolutional network is essentially another type of neural network which is a very important type and has been profusely used in drug discovery and in life sciences. In the convolution, we normally handle images, a kernel or a filter. Multiple filters are used to convolve a local region and produce future maps by using taking the dot products of the local regions and then producing future maps after activation. So multiple future maps result from local region mapping, convolving, and after several filters depending upon the particular problems. You can use different types of filters depending upon the big data handling capabilities and the particular problem and image intensities and number of images at hand. Once you get these future maps, we can reduce the dimensionality to reduce the overload of data by using some sort of pooling methodologies. Once you do this, this can be done for a few number of layers until the final layer where you have a fully connected layer, and this fully connected layer is now activated and sent to the output. And like usual neural networks, the prediction errors are backpropagated to readjust the weights, and the process is iteratively done to minimize the error. LSTM is long short-term memory neural networks, deep neural network methodology, which uses a series of these cells, and it uses using the cell state information to carry the informative information from the previous states and to discard useless information. So this LSTM is essentially used to preserve the sequence information. Like in the earlier example, we had given set of protein sequences. We extracted the amino acid frequencies. Now, different sequences may have the same amino acid frequencies, but by extracting the amino acid frequencies, we'll use the sequence information. A particular sequence, pattern of sequences, which is now, you can use this sequence information, and the sequence information can be preserved in this series of LSTMs, and which can be employed for high performance drug discovery problems. Yeah. In this paper, they've used X-ray images of COVID-19 with a combination of deep neural networks, both CNN and LSTM, for detection of coronavirus. Yeah. So the input data consists of different sets of X-ray images. It contains 3 different classes of images: pneumonia, COVID, and normal images. Uses some certain data pre-processing methodologies, and then converts into training and test data. The training data is used to build a CNN LSTM combination model, and the test data is used to test the model parameters and identify the most optimal parameters, and finally get the model. And then the model is evaluated by using performance measures, and this process is done until you get the best set of parameters. Yeah. So the X-ray image is passed to a set of CNN layers. The output of the fully connected CNN layer is now sent to a series of LSTM cells, and finally you get the fully connected layer, and output is now predicted as normal X-ray or COVID image or pneumonia image. Yeah. So we have so far talked about different kinds of descriptors. We have used different omics data like sequence data, structural data, and physicochemical properties, and then multiple types of omics attributes. And then we have explained a few methodologies for certain examples like antimicrobial peptides, prediction of solubility, etc., and gene finding. So these sort of attributes, omics attributes, can be used for certain set of applications. Now, the second set of attributes are imaging attributes, which can be MRI images, radiographic images, and histopathology images. And then it can also be, as I said, X-ray type of images. You can use these images, the image applications, as we have just talked about, applications in COVID identification. It can also be used for brain decoding and other methodologies, other particular disease predictions. The third type of images is biomedical signals. They are not images, biomedical signals. So you can extract time domain attributes, frequency domain attributes, and multiple attributes from this, and then use these attributes as input to the machine learning methodologies and identify multiple diseases. Yeah, this is just a cartoon of different papers which have appeared recently on different methodologies. Yeah. Quite early, we have employed the arrhythmia, classified arrhythmia using wavelet domain attributes. In this, we have 2 classes of ECG signals, and then we have large number of examples belong to 2 different classes of signals. What we did was, from this direct, because the time domain contains, for a very small amount of time, large amount of time domain data, which is difficult for us to use the time and domain attributes to to rationally classify the images into different groups. What we did was we took it as a wavelet domain, and then from the wavelet maxima region, we further extracted some attributes and used those small number of attributes with multiple time series signals to classify the ECG signals into different groups. Yeah. The third type of learning is reinforcement learning. In reinforcement learning, as I said, we learn from experience. It uses rewards and penalization to train reinforcement learning models to perform a sequence of selections. And it learns from experience the multiple levels of passing through different reward penalization states. Finally, it tries to achieve its job. One such application is a deep reinforcement learning algorithm used in conjunction with the CNNs and deep generative network, which is known as GAN. Yeah. So starting from noise signals, it's possible to generate certain molecules. These molecules which are generated have been passed into the CNN networks, which employs a supervisory learning methodology of predicting from the set of generated images the probability of these generated sequences to have specific drug activities against certain diseases or not. Using this information, the deep RL rewards or penalizes the set of sequences and sends this information to the GAN network, which further improves and then produces better molecules, and the process goes on until we get a set of best possible de novo molecules with having specific physicochemical properties. Yeah. So I think we have a few other applications are also there. You can leverage large amount of electronic health records and then use those digital health records combined with genomic information like the presence or absence of genes and mutations and single nucleotide polymorphism variants, genetic variants, information from jivas, and then our methodologies to identify disease etiology better. ML models trained on the electronic health records recently have enabled mortality risk prediction in COVID-19 patients. A large amount of data is there, so we need to have big data handling methodologies with good enough computing power. And essentially, you only can use deep learning methodologies for these approaches. Yeah. And they also have been used in precision medicine. Now, patients can be personalized by demography, medications, comorbidities, their disease trajectories, and genetics. Yeah, because of sequencing in reasons, personal genomes can be sequenced for less than $200 nowadays. The genetic jivas information can be used for individuals, and its multi-omic profiles and molecular data can be used, and this information can be used to prescribe precise medicines for personalized treatments. Yeah. Now, several startups have already started creating biomarkers for cancer therapeutics. And this particular ASTEC is specializing in providing precision medicine software tools for supportive care. The Precom Therapeutics is providing drug profiling solutions for personalized medicine. Yeah. Machine learning based methodologies can also be employed for drug repurposing. In drug repurposing, we can use the same drug target. We can use multiple therapeutic uses we can have. Or we can, for a given drug target with the same drug, for the same drug, we can identify different drug targets for repositioning drugs. Now, a recent example is the computational identification of several antiviral drugs, including remdesivir, and this has been essentially made possible by machine learning studies. Yeah. Now, the other applications is in psychiatry and mental illnesses. So, you identify mental illnesses by using different imaging methodologies. Yeah, you can also use to get insights in social behavior and personality traits of people. You can also identify traits of people who have particular sets of diseases, psychological diseases, and then timely interventions can be made by using these methodologies. Yeah. Now, machine learning has been used in animal behavior to track animal movements in parks, and also been used to monitor endangered species by using imaging, and then using sensor methodologies to count the number of species by using a remote identification by using wireless sensor networks. In dairy industry, animal motor sensors can be used to track the activities of cattle around, and also for several other applications in animal behavior we can use. In plant genomics and plant breeding, you can use different genetic variants to get the good varieties of plants, and then we can also use different culture medium, tissue culture. So the fermenter we can use cultures in laboratory using fermenters to a priori identify offline experimentology to optimize the culture mediums using different plant growth regulators, etc., and identify different media concentrations, which best gives you the proper yields of the required material. Yeah. Similarly, it can also be used in breeding of animals in dairy industry. So cell types identification is a very important area in biological research. Now, cell types can be used by different image handling methodologies like segmentation and other techniques, and then identify images of different types of cells, and then which can be used in classification of diseases. Yeah. Can also be used in public health, in prevention of diseases. Deep learning has been used in applied in monitoring and predicting the concentration of air pollutants, tracking disease outbreaks, and modeling disease progression. Recently, it has been used for COVID modeling at different locations by using time series data. So time series data handling in machine learning itself is a specific field of study, which is different from normal machine learning methodologies. Yeah. Finally, the machine learning can be used in industrial fermentation products because different fermentation conditions have to be used for producing different products. So the design of fermenters or design of the fermentation media and operation, etc., ML can be used at different stages of fermentation processes. Yeah. I think with this, because I will now stop my lecture on this applications of machine learning to life sciences. Although I have listed a list of subfields where ML can be applied in life sciences, there can be multiple other subfields which I might have missed or I don't have time to. So I think you can use your own discretion to learn more subfields where ML can be applied. I think so. The applications are limited to whatever I have talked. There are multiple applications which I have not given. So, with this, I will now stop my webinar lecture. I am ready for answering questions. Yeah.
Thank you, Dr. Jayaraman, for your wonderful presentation, giving a very comprehensive introduction to the complex subjects, both the machine learning as well as life sciences. For many people, both are complex, but you have given a overview so simple, and then given a list of applications, the array of applications which no one can think of, in such a short period of 1 hour. You have covered a lot. So, in fact, it's really overwhelming for many of us, because quite a few of us are new to life sciences. Basically, the target audience, who are present in this webinar, they're all from computer science background, and life sciences, they do find it a bit difficult to understand. So let me start with a first question. What type of subjects one has to learn in terms of using the machine learning? Basically, this is with reference to the computer science students, with reference to computer science students, what type of biology related subjects, one has to get familiarized so that he can apply the knowledge of computer science?
See, the applications of computer science methodology. Yeah, yes, sir. Something else?
Yeah, can you come on video, sir? Can you just put your video on?
Yeah. I will come on video. Yeah. Yeah, I will stop sharing.
Yeah, you can stop sharing and you can be on the video.
Okay. See, essentially, computer science, where machine learning has to be applied, people have to understand different domains, not only life sciences. So they must be prepared to learn the domain, multiple domains, and including life sciences. Have to start working on life sciences and computation biology. They must start learning the bioinformatics principles. The bioinformatics is a sort of midway between informatics and the biology. So they must start learning about the genomics, about genes and protein sequences, and start using simple set of attributes, simple set of problems. So it's, see, I don't believe in learning a complete domain and then start applying it. So you must have a good expertise in your own area. If you have an expertise in coding, just take small problems with the help of some person, some guru, and then start learning the domain slowly. As you apply, another way of doing it is you learn from available online platforms like Kaggle, where large number of problems in different domains are solved. The programs are available, the methodologies are available. So you get, once you, from the description of the data sets, what sort of attributes have been extracted. Like, for example, in the protein function identification problem, you can use simple attributes of 20 different amino acid sequences to classify certain protein functions with very good enough accuracy. So you start with simple applications, try to run the applications yourself. As you run the applications, and then more and more problems you can take. So it's a gradual learning curve. And then, as I said, if you know somebody you can teach along with the learning of the particular problems at hand, maybe a small short course you can take in bioinformatics, and then, and as I said, you cannot learn something completely and start working on it. It has to be slow and steady, and you must solve practical problems using these areas. Yeah.
Yeah, okay. With this life sciences, basically they generate a lot of DNA sequencing and others, generate a lot of data.
Yeah.
So, which means that processing power is one of the main requirement. So, what type of computing power is required for this analysis?
See, it depends upon the particular problems. For normal omics-like problems, you can use many problems we have solved by using a simple computer with small 8 and 16 GB this thing memory. But you have to handle images and multi-omics data. You may have to go for big data handling methodologies. You may have to go for some supercomputers. Yeah.
Okay. And can you give some examples of genetic algorithms, or genetic programming being used in life sciences?
Yes, yes. See, the genetic programming and genetic algorithms have been used to identify the most important attributes. What is we call it as future selection. Now, the future selection, as I said, can come from domain information. But if you've got large amount of domain descriptors, it is very difficult for us to identify the attributes which are important. So, GA as a methodology, you can start with the genetic algorithms and genetic programming and use different initial trial populations of subsets of most important attributes. Iteratively, you can arrive at the most important attributes. And yeah,
Can you suggest a segmentation technique?
sir, there are multiple segmentation techniques available. And yeah,
Okay.
yeah.
There is a on generic question. Will machine learning help human race advance to its next level, or is just a hype?
I think machine learning has indeed helped us advancing to other levels of. We have got increased healthcare facilities by using machine learning methodologies. E-commerce uses machine learning profusely. Lifestyle has improved profusely. So I think what is the next level definition is important. In our opinion, machine learning has definitely helped in progressively improving. What people say, the next generation of people may not learn driving at all because autonomous cars are going to be used. Yeah.
Are there any major failures of applications of AI in life sciences?
Um, AI has failed in multiple drug trials. It has failed, and then companies will not
Disclose it.
disclose it. That is one. But there's a IBM University of Texas, Texas and Austin combination where they have tried to get expert system for oncology, and very bad recommendations were made, and then it is a major failure. Yeah.
Okay. And there's appears to be a little trivial, but what is the difference between machine vision and machine learning?
I think the machine vision as a computer vision is just using digital images to recognize objects, object tracking and movement. Yeah, there is no difference. Actually, machine vision and is again uses machine learning methodologies like deep learning, track objects and detect digital images. Yeah, it's a handling of digital images. See, we have our eyes we use years of context available to track different images, the distances of images, closeness or distances. But we have to use computer vision methodologies with cameras and algorithms and very fast to identify these things. Yeah.
What type of validation techniques are employed to justify the outcomes of machine learning?
So what did you say? What sort of?
Validation, experimental validation. Is it being done based on the machine learning outcome?
Yes, sir. See, machine learning by itself is a prediction methodology. See, the prediction, although many methods claim that we have 100% accuracy or more than 95% accuracy, it may work on the training data, but on the new set of data, many methods fail. So we have to use the methodologies during algorithm training. We have to use statistically unbiased method of preparing algorithms. Once you do that, there are ways of predicting, even if a particular, for a given example, if we identify a protein which is going to be soluble, now we can use the probability of this protein to be soluble can also be computed by machine learning methodologies. So the best way is to take the highly probable predictions and experimentally annotate, and then that experimental annotation of these highly probable proteins will tell you how effective your algorithm is. Yeah.
Okay.
And you can add these highly probable sequences into your algorithm, again retrain, and then the procedure is again sequential. Yeah.
Okay. See, basically for machine learning, the quality of data is very important, right?
Quality and the amount of data. More the
Amount
data,
of
the
data.
better.
Yeah, quality and amount of data.
Yeah.
But unfortunately, country like India, where medical records and health records are not digitized. So what way that we will overcome this problem?
It only has to see large companies, and then government has to come forward to prioritize this digitization along with enabling hospitals, to do this. So it's a characteristic methodology has to be used for digitization, and this is something which is urgent need of the hour.
No, so which ultimately means that the biodiversity of country like India is quite different from the Western countries. So
Correct.
which means whatever studies done in Western countries may not be applicable to Indian continent, right?
Most of the methodologies are specific to regions. Yeah.
Okay.
See,
So
even the personalized medicines,
yes.
now the genetic variants are also different. Now we know the COVID, even in India, you see, the Kerala has got more COVID cases than other cases. Yeah.
So which means that we
We
are
cannot have a general uniform model for prediction for COVID in India completely.
yes.
We will fail miserably. Yeah.
So ultimately, this means that we are quite far away from personalized medicine, right? Or
Yeah, we are quite far away. The personalized medicine is currently for very rich people. So for very rich people, you can get multiple molecular profilings and then genetic variant analysis, and then there are ways of getting enriched data for people with multiple methodologies, and we can use algorithms, and then with some success they're used. Yeah, but it is out of reach of common person. Yeah.
okay, now there had been press reports about DNA profiling is going to be less than $100 and things like that. Has it become reality, or is it still a distant?
Yeah, DNA profiling definitely is genome profiling is around $200 in US now. Yeah.
Okay, so but basically there are some ethical issues in terms of DNA profiling, right? So people feel that DNA profiling has got some negative impacts, ethical issues. What is your opinion on that?
Yeah. So everywhere the ethical issues have to be taken care of before profiling. So but in India, this this itself, as you said, is a distant dream. But ethical conditions have to be very strictly adhered to. Otherwise, it will end up in misusing. Yeah.
And can you throw some light on DNA storage?
DNA storage. I have not worked on that, sir. Not much I know. Yeah.
Yeah, because now they claim that DNA can store
Yeah,
a
but
huge amount of data.
They say, but this has been
Practical,
IBM
practical
also has.
application. Yeah.
Yeah,
Okay. Yeah.
IBM has claimed they have developed a methodology, but we do not have any evidence of this being used in practice. Yeah.
Now the big tech companies like what IBM, they have come with Watson, and then Google, they started life sciences research and things like that. So,
Yeah.
so this means that this domain is moving away from the conventional biologists to computer scientists.
I don't think a computer scientist from different domain can completely understand the biology. I have learned biology myself, but I have not come to a stage of knowing the intricacies of biology fully well. So it has to be a mix of people with different expertises together to work so that you achieve the end goal. Yeah.
Okay. Then the one last question, sir. In terms of now there are a lot of variables of I mean like Fitbit and Apple Watch and things like that. So these are going to help in terms of data collection, and so that will possibly will try to get some unadulterated data, right? So that will possibly will help us in the research.
Yeah. See, this is sort of smart sensors we have,
Yes.
and smart sensors can definitely collect data which can improve our lifestyles, essentially improving our diet, then our exercise behavior and fitness and all. So essentially, in fitness, it's going to be quite helpful. Yeah.
So thank you very much, sir, for your excellent presentation. And now I request Dr. Sakthivel, the chairman of IEEE Computer Society, to propose a formal vote of thanks. Dr. Sakthivel, you can unmute. Sakthivel.
Sorry, sir. So good evening to all of you. On behalf of the IEEE Computer Society Madras chapter, ACM Chennai Professional chapter, and the Computer Society of India Chennai chapter, I would like to thank today's resource person of our webinar on applications of machine learning in life sciences. So machine learning is an emerging area, and our resource person, Dr. V.K. Jayaraman, has explained in an excellent manner. It is a very comprehensive one, and all the participants of this webinar, I hope they have benefited from this lecture. Thank you very much from Dr. Jayaraman, sir. And I would like to thank Mr. H.R. Mohan, Chairman, ACM Chennai Chapter, Dr. Priya Vijay, Chairman, CSI Chennai Chapter, Mr. P.V. Subramanian, Past Chairman, CSI Chennai Chapter, and Dr. Ahila, Managing Committee Member, CSI Chennai Chapter, for their excellent support in organizing this webinar. And I would like to thank all the participants who have joined through this virtual environment and their active participation through various questions. Thank you, Ananda. And now I would like to inform some of the forthcoming events organized by these societies. On 9th October 2021, we have a webinar on radical transformation in data analytics. And on 23rd October, we have a webinar on best practices for commercially successful patents. So the circulars will be sent to you in due course of time. And I request all the participants to register for these webinars and attend it and get the benefit out of it. Thank you, Ananda. Thank you.
Thank you, Dr. Sakthivel. And sir, once again, our sincere thanks to Dr. Jayaraman for his time and effort in presenting a wonderful seminar, workshop. And I can say, it is much more than the workshop. I can say it's the 1 hour input, what we have received is something beyond our imagination. So thank you very much for your time and effort. So we really look forward for a longer session. Possibly, each aspect can be dealt in more than, it may require minimum 15-20 minutes so that to go into the details. So definitely the 1 hour time what we are given is we are not done justice. Possibly we should plan a 1-day workshop or some detailed program. Hopefully we can approach you for at a later date now to organize this program, sir.
Thank you very
Yes,
much, sir. Thank you very
very
much.
true.
Yeah, okay, I will wait. Yeah, I will wait till
Yeah.
others talk. Yeah.
Yeah, there are someone is
Yes,
someone.
yes, sir. I'm Priya Vijay, sir. Yes, sir. It's very true, sir. We can organize a workshop. We gave lot of applications, which because now all our research people are moving towards only medical field, which helps to publish paper also soon. So all are moving towards only medical field. So we can conduct a workshop so that
Okay.
lot more will be benefited.
Yeah, for each application there can be multiple
Yes.
papers. There are
Yes.
so much is there. So definitely,
He gave lot of applications. Lot of
yeah.
applications.
I
Thank
mean,
you, sir.
entire,
Thank you.
I think entire cross spectrum of applications. Right
Yes.
from plant sciences, animal sciences, health. Human science, everything, all the aspects. So it's like an encyclopedic presentation, I think.
So I thank Professor Mohan and all other executives and all IEEE and associated agencies for giving me an opportunity to talk. And then it has been a wonderful arrangement and dot on time. And then the management has been right from the beginning has been excellent. And our interactions, I think, is quite fruitful. And we'll continue this interaction for a longer time. Thank you, sir. Yeah.
Thank you very much.
Thank
Sir.
you. Thank you all.
So if any audience want to ask questions or interact with the speaker, please do so. Now it's open.